Sir, is my voice is audible?
Yes, your voice is audible.
Yes, sir. Yes. Good morning, everyone. We will start our session soon once all the participants are joining. We'll wait for some time and we will start our session. an announcement to all the registered participants to use their registered name in their account. So there was like different names instead of the registered name, so it is automatically recording the attendance. So it is mandatory to provide your registered name for this Zoom link.
Okay. So,
Good morning, everyone. There is an announcement. So, all the registered participants are to make their names as per the registered in the form. So, please rename yourself in the Zoom meeting since the attendance is taking, it is taking automatically. So, registered participants are requested to rename themselves. We will start our session. Warm good morning to everyone. A cheerful welcome to all the participants, respected speakers, faculty members, and the organizer to the day 2 of our workshop. I hope all you had an insightful and engaging day 1 filled with a valuable learning and discussions. Today, we have another exciting lineup of our expert sessions focusing on advanced analytical techniques, both chromatographic and spectroscopic, which plays a crucial role in modern research and product analysis. To begin our day, we have an enlightening session on chromatographic techniques, a powerful tool for food processing research. We are privileged to have Dr. Vivek Kamambati sir, an expert in this field, to guide us through the fundamentals, advancements, and real-time applications of chromatography. Sir is currently serving as an assistant professor in Department of Food Process Engineering at the National Institute of Technology, Rourkela. His research of expertise include process modeling and optimization, food safety and quality management, functional foods, drying and dehydration, novel and non-thermal technologies, post-harvest management. He holds a PhD in food process engineering from NIT Rourkela. Sir has published extensively in reputed journals with over 500 citations with an X index of 12 and active serves as a reviewer for several high impact scientific journals. Thank you, sir, for being with us. The session is now over to you.
thank you, Divya. Thank you for your introduction. So, good morning, participants. Today is the day 2 of the workshop. Yesterday, you had a session on importance of food analysis in food industries, and then you in that particular lecture you learned about what are the different analytical tools and what are the different analytical techniques are being considered in the food industry. So in the second lecture, you learned about the sampling techniques, right? The sampling techniques and some sample preparation, how to prepare the sample. If your sample is wet sample or if your sample is dry sample, how to prepare according to the FSSAI norms, you have learned that one. In the third lecture, you had a very good, interactive session on this practical demonstration of proximate analysis, and you learned about different proximate micro and macro nutrient analysis and how to interpret the results, how to calculate the results. Those things you have learned. So today it is little bit advanced. Okay, yesterday was the very basic level. Today one step ahead, right? So today you will be learning about the chromatographic techniques. So we'll learn about what are the different chromatographic techniques available, and then in the second lecture you will be learning about the spectroscopic techniques. Okay, so without delay, I will just share my screen. Divya, can you tell me, is the screen visible?
Yes, sir, I can tell you. Yes, sir, the screen is visible.
Yes. Is it moving to different slides?
Yes, it's moving,
Yes.
sir.
Thank you, Divya. Thank you. So I will be covering this topic, chromatographic techniques in food analysis. So these are the learning objectives. So what you will be learning. So after learning, what you will get from this particular lecture, you will understand the principles of the chromatography, and then you will be able to classify different chromatographic techniques, right? How many different techniques are there, and you will know which technique has to be used for what application, right? Then you will learn about how to identify the application in food analysis, how to use this particular chromatography techniques in a different application of food analysis, and we'll discuss some of the case studies related to food industry. Okay, so before entering into the advanced level, I would like to start from the beginning about what is chromatography. This is small introduction about the chromatography. What do you mean by chromatography? Okay, chroma means color, color grading. Okay, chromatography means based on the colors you will identify, you will read different analytical substances present in your food material. This is an analytical technique for separating the mixture. Chromatography altogether combined is a separating mixture. It is an analytical techniques. So in this particular method, you have two different phases. Okay, the first one is stationary phase. So you can see in this particular image, you have mobile phase either in the form of water or any kind of solvent. You have a slide. Okay, on that, if you just point, okay, if you have a marker or any kind of color, you just make a dot in it and just put that slide in the water. What will happen? It is not a slide, it's a paper, right? If you put it in the water, the color, okay, water slowly it will start migrating to the top portion, right? It will just travel towards the top. As the solvent travels towards the top, you can see different colors. Okay, this is the indication of different compounds present in your sample. Okay, based on the color, you can easily get the graphy. That's why it is called as chromatography. Okay, this mobile phase, it will be traveling, and stationary phase, the paper or slide, it is stationary and it contains the sample in it. Okay, based on the differential partitioning between the phases, you can identify what are the compounds present in the sample. Some historical background you can see. This particular method is invented in the year 1903 by Michael Sweat. He is the one who discovered the principles of chromatography using the plant pigments. He analyzed, taking a plant, any kind of plant, and then he identified different pigments present in the plant: xanthophylls, chlorophylls. This is the first, you can see in this figure, A is the first chromatography made by this Michael in the year 1903. the color writing. He already gave the name as the color writing. Then slowly the HPLC, okay, high pressure chromatography, based on the different mobile faces, okay, different applications, the chromatographic tools and equipments have evolved based on the different needs. But this is the initial setup. You can see in this particular jar the mobile face will be filled, okay, and then the mobile face along with the sample, it will be traveled inside this column, and then it will come on the stationary phase. This particular pipe, it will be connected onto this stationary phase. In the stationary phase, different solid materials will be packed, okay, in this tube. Based on the color, the separation will take place, right? As the solvent moves, it will be dripping into this conical flask. The color separation will be there. That's how the principles of chromatography have been evolved. The general principle is like this. In the modern labs, or in your industries, or maybe in your the institutes, you can see this kind of setup. Okay, normally you have a mobile phase, and then this particular mobile phase, any kind of solvent, it will be pumped using the motor, then it will be sent into the column. Am I right? The column, all these things we will be learning in the next coming slides. What are the different columns are available and where we can use all these things. And then you will insert. I request participants to mute your audio. Okay, you will be injecting the sample, and then this mobile phase will take the sample into the column. Because of the temperature difference and then pressure, okay, what will happen? Separation will be taking place. Okay, based on the separation, the detector will be detecting how many different types of molecules are present, and it will give it in a peak in your it is called as the chromatogram. We'll also try to learn about what is chromatogram. Then that particular, after separation, then the wastewater will be released along with the sample. Okay, whatever materials not absorbed into the column, it will be sent outside. Okay, that is called as the waste. Compounds separate due to the differing affinities. Okay, what do you mean by affinities here? The column will be decided based on what kind of compound you are targeting. Okay, if you see any kind of research paper, different columns will be mentioned, right? This particular column, this is the length of the column, specifications will be given. So that particular column can be used for analyzing some kind of food materials. Okay, it cannot be used for all the kind of analysis. Okay, we'll try to learn that one as well. Then elution profile represents as the chromatogram. Based on the peak, the area, we can easily identify how much quantity of the target compound is present in our sample. We can easily identify. One good example I will tell you, the estimation of sugars using HPLC, right? You can analyze glucose, fructose, but in the case of sucrose, okay, it's made up of 2 monosaccharides, right? Again, you need to read how much sucrose and how much fructose is peaks are coming, and you have to calculate the peak. Accordingly, you need to use the standards. Okay, so these are the classifications of chromatography. In 1903, it started with only mobile phase, and it started with liquid, mobile phase, and the solid phase as the different adsorbent solvents. Later, it got evolved into gas chromatography. The solvents or maybe mobile phase can be different, right? Different researchers, they have identified, okay, we can use gas for separating or adsorbing some kind of components. Then liquid chromatography, and then supercritical fluid chromatography. Under you have gas chromatography, gas liquid chromatography, gas solid chromatography. Okay, the setup will be in the column. Okay, in the liquid chromatography, you have ion exchange chromatography, exclusion chromatography, partial chromatography, and liquid solid absorption chromatography. This liquid solid absorption chromatography is also called as the thin layer chromatography. As you already heard about this thin layer chromatography, we'll learn those things as well, right? These are some of the key components of chromatographic system. You take any kind of chromatographic system, either it may be HPLC, GCMS, or UHPLC, or any kind of chromatography. You can find these components. These are very important components. You can find solvent, okay, either in the form of liquid or gas or supercritical liquid, and then you can definitely find a stationary phase, right? The solid or liquid coated stationary phase, where which your target component will be absorbed and it will be identified by the detector. Then you have a sample injector. Okay, sample injector, you have to inject the sample, then it will travel long way inside the column before it hits the detector. Okay, then data processing using the computer software. This chromatographic interpretation, you can see you have three different things here. You can see in this particular figure, you will get a graph like this. Okay, what is the meaning, how to read the graph? So in the practical session, it will be shown clearly. This particular peak, what is the height of the peak, what is the area of the peak, is very much important. And one more important thing is retention time, when you are getting the peak, right? That is very much important, because see, with respect to time, in the HPLC, I will tell you one example. If you are identifying the sugars, maybe sucrose, for example, it will start showing its peak after 4 minutes. Okay, if the peak is coming before 4 minutes, then your sample, there is something wrong in your sample. How can you identify, how can you interpret that one? By reading the standard first, pure glucose standard, pure fructose standard you need to use, and then you need to obtain the curves. Okay, then you can correlate with your sample. If your sample is a fruit juice, then you will inject that one in the HPLC. What it will show? It will show a peak after 4 minutes. Then you can compare that peak with the standard peak. Okay, what is the area of the standard peak after 4 minutes? At that particular time, this peak will start showing, right? What is height of the peak? Based on that, you can calculate how much quantity of the sucrose or glucose or fructose is present in our sample. That is called as the retention time. It is denoted by TR or TN. Okay, it is a time to elute components. Okay, the component will start showing peak area proportional to concentration. See here, definitely, if the concentration, if you are using 100% glucose, okay, diluted in 10 ml, it will show strong peak. The same 100 ppm of glucose, if you are diluting in 200 ml of water or 100 ml of water or solvent, the peak will not be very strong. It is very short peak, you can observe, right? That's why the peak area is proportional to the concentration. Resolution is a clarity of the picture and clarity of the peak. Okay, if there is no rigid or bumpy peaks, then you can say that your identification is right. If you have a some kind of a rough peak, then there is some error occurred in your analytical method. Maybe sample preparation is wrong, maybe some the instrument wise maintenance or calibration, something is wrong. The peak that you should get is very smooth peak at a particular retention time. This is very important. Okay, there is also one more time involved here. It is called as T0. It is called dead time, where which the instrument will undergo processing for 20 minutes, or maybe for making the machine ready, we need to set some time, right? That is called as the dead time, after which your real analysis will be starting. After that dead time only, you will get the retention time for any kind of this one analysis. So this is how you'll get, this is an example I've showed here. This is the UPLC profiling of plant pigments. So how you can read this one? This is the time, okay, versus relative intensity. I told you peak length is directly proportional to the intensity of the materials present in the sample. Am I right? Strong peak, high peak represent high concentration of that particular pigment present in your sample. Okay, and lower peaks, you can see here this peak is very rough, okay, and very low. This is not at all counted. This is not, we should not identify otherwise we should not consider this peak. If you are going for the profiling or fingerprinting, then you can identify how many peaks are there, and these peaks correspond to what kind of material. Okay, but if you are going for specific compound like chlorophyll A, estimation of chlorophyll A in your sample, okay, maybe plant sample or whatever sample you take, then you will get very good. The retention time is around 5.8 or 5.7. okay, from where you can get this retention time. This retention time you will be knowing by standard. Okay, chlorophyll A standard you will run, you will identify sorry, you will identify the where is the retention time for this particular chlorophyll A. Okay, and then see in this particular figure, in the standard analysis, you got retention time just near your 5.7, 5.8, you get two different curves, but the length of the the peak or the area is less in 20. Both are chlorophylls, but here you see the first peak is high and then second peak is lower compared to the 19th. Why? Say both are chlorophylls, but why there is a difference? Okay, how to interpret this one? Peak 19, this particular peak is very strong. No, lot of high concentration of that particular pigment is present in the sample, corresponds to the major native form of the chlorophyll A. Okay, but whereas in the case peak 20, chlorophyll A likely represent a slightly modified or isomeric form. What do you mean by isomeric form here? Isomeric forms are compounds with the same molecular formula. Okay, what is the molecular formula of chlorophyll A? And what the same molecular formula it will be having for the different isomeric components, right? But a different arrangements of atoms in it. Okay, leading to different chemical structures and physical properties. That's why you can see the molecular structure formula is same, but the atom arrangement is different. Okay, that's why this is reading it as a chlorophyll, but 19 is the correct one. 20 is the modified form of the chlorophyll. Okay, the structure it is given like that, but it is slightly modified isomeric form. It can be epimeric form, solvent associated complex, or maybe early stage degradation product. Okay, when the chlorophyll is degrading, then that particular degradation will change the atomic arrangements in the chlorophyll, right? That particular pigment. So that may be shown in 20. Like this, we need to identify what is happening, where we are getting the pure peak, where is the highest length of the peak, and is there any degradation. See how you can identify the degradation. Slightly, chlorophyll, a pure extract you will keep, and then after 10 hours or after 12 hours after degradation, you can keep, you can read this particular UPLC profiling by keeping that particular chlorophyll, whatever the target compound, for different periods of time. Okay, you can find out the kinetics, how the chlorophyll is degrading, degradation kinetics of chlorophyll, that you can easily get from this UPLC. Okay, then the factors affecting the separation process. What are the factors affecting separation process? Definitely two things are very much important. Stationary phase and mobile phase are very much important. You need to identify the suitable mobile phase and suitable stationary phase. Otherwise, whatever sample you will analyze, it is waste. Okay, you will not get a true results. Right. That's why nature of the stationary phase is very much important. Mobile phase composition and flow rate is also very much important. If the flow rate is too high, what will happen? The absorption time required for adsorbance of that particular component will not be sufficient. Before absorbing 100%, it will just flow towards the waste. That's why the flow rate is very much important. Then temperature in the the column is also very much important, because with respect to temperature, the component separation will be there. All these chromatographic principles are based on the separation, right? Separation of your different components. Column length and particle size is also very much important in case of chromatography. See here, I would like to ask you people, is there any ideal peak shape in the chromatography? Any one of you can answer. Is there any ideal peak shape in the chromatography? Yes?
No,
Anyone?
sir.
How?
First,
Sir, your voice is not audible, sir. Hello, sir.
first one, sir.
Sir?
First peak is ideal.
Yes, yes. You will get different types of peaks, right? So which peak you need to consider is very much important. As I told in the earlier slide, the peak should be smooth without any rigid surfaces on this one, right? The symmetric peak is ideal, but in practical cases this is not possible. You will get fronting peak or tailing peak or some kind of rigid appearance like this. You can see here in this picture, they see this is not smooth, right? See here, lot of disturbances are there in the peak, but see in this peak it's almost smooth, but some some corrections are there, but still it is not smooth, right? In the practical case you will not get. But see this peak number 3, number 4, all these things are very zigzag, right? See here, this peak is having a very small projection here, then high projection like this. But this particular peak at this particular retention time has a single peak, and it's almost smooth compared to other peaks. Can you identify here? So that's why these kind of peaks you need to consider for the analysis. That peak called as the symmetric peak. Symmetric peak means what? If you divide that peak into two different sections, then area A and area B should be same. A should be equal to B. In the fronting peaks, A is a little greater than B. The area of A is little greater than B. Whereas in the tailing peak, it's reverse. Okay, the peak tails backwards, right? So these kind of peaks are possible. You see here, is there any ideal peak? Yes. The ideal chromatographic peak is symmetrical. It's a Gaussian shaped peak. The left half should be equal to the right half. It indicates uniform interaction between the analyte, stationary phase and mobile phases. Where you will get this peak? This peak is possible for the standards that you are inserting in the HPLC or maybe GCMI or any kind of chromatography. You will get a very good smooth peak. Okay, but again, resolution is also very much important. What is the clarity of the peak that you are getting, right? So based on that, you can identify symmetric peak means what? Proper sample loading. Okay, you properly loaded the sample, the required quantity of the sample is loaded. Stable flow rate, you optimize the flow rate. Okay, 0.1 ml per hour or maybe 0.2 ml per hour, you optimize the flow rate. And column settings are very good, no secondary interactions are possible, right? You washed your entire equipment inside the column thoroughly. You have to for rinsing, right, before inserting the sample. So that part is clear and there is no secondary interactions are happening. Okay, and then no overloading or active sites on the stationary phase. Okay, this is the meaning of the symmetric peak. Okay, but in practical cases, what will happen? We will not get 95% will not get the Gaussian peaks. Okay, symmetric peak is not possible. Some slight deviations will be there. Okay, so those peaks will fall under two different categories, either Gaussian, tailing, or this A is greater than B, fronting or tailing peaks. What are the causes? You can learn here. Column overloading. Okay, if the area of A, the peak front, means column overloading, too much sample is injected. Column overloading means instead of inserting 0.5 ml, you inserted 0.6 ml or 0.8 ml. So how it will be possible? Okay, if you are not properly calibrating your the injection, or maybe if you're not properly calibrating the whatever the material you are using for taking out the sample, then this problem may occur. Column overloading will occur. Then the strong adsorption of annulate to stationary phase. Okay, so this is also possible, right? In the stationary phase, you will have stationary phase, right? So there is a strong adsorption, okay, of annulates. Whatever the compound, target compound, it is strongly absorbing. Okay, that's why the peak will be little bit tilting. Okay, too fast flow rate or mobile phase incompatibility. This is very much important. Annulate travels faster than expected. Early part of peak will be sharper. Okay, this is the meaning of the A is greater than B. What happens if A is less than B? What are the causes? Secondary interactions. What is secondary? With active sites on the stationary phase, ionic or hydrogen bond interactions will be there. Okay, so this kind of secondary interactions we need to avoid. Otherwise, you will get a peak tailing. Contaminated column. Maybe column is not properly washed, right? So you did not give it for a rinse before injecting the sample. Improper pH. Okay, the sample pH is also very much important, right? So if you are instead of 5.5 or 6 pH, you are give the pH of the sample is a little less, maybe 3.5 or 4, then there is a chances of secondary interactions. Okay, the column is made up of metal, right? So these kind of things we need to avoid. We need to identify what is the optimum condition for our sample. So that's why if you see in the research papers, even though the chlorophyll, example I'm telling, chlorophyll is identified, in each and every research paper, they will give the method, right? This much sample is taken, this is the flow rate, this is the column condition, and this is the specification of the equipment. But you can see in different papers with slight modification. That particular method was not working for the other scientists. Okay, they are seeing some kind of disturbances in the sample analysis. That's why what they will do, they will identify what is causing wrong in analyzing that particular sample. So they will change it little bit, and they will enter those specification in their research articles by mentioning slight modifications. Instead of 0.5 ml per hour, they used 0.51 or 52 ml per hour, so that they get the good curve, smooth symmetric curves without any kind of deviation in the curve areas, right? So the next possible cause may be detector overload. Okay, so detector is also very much important, right? We'll learn about those detectors. So what is the meaning of A is less than B? Some analyte molecule gets retained longer due to these interactions, secondary interactions. First is adsorbent molecule, analytes travel faster than expected. So this is the main difference between the peakings. This is possible in most 90% of all our analysis. We do analysis, but we do not know whether our results are correct or not. So you need to go for the standardization of your procedure, or maybe standardization of your the sample take sampling process. That's why these two things are very much important. This NABL accredited laboratories, or the commercial laboratories, they do standardization quite often. But in normal labs, we analyze standards once in a while. We use the same graphs. Okay, we sometimes we fail to calibrate the equipments. Even the weighing balance calibration is also very much important, right? So that's why it's very much important to know what type of materials we are using before the analysis. Okay, fronting or tailing, when is it a problem? You can see here, fronting, minor slight tilt is acceptable. Severe distortion problem may cause overlapping with the next peaks. Okay, in this picture I showed you, right? This is the overlapping. Some overlapping is happening, so you cannot read this as a pure curves. Okay, some overlapping, two figures, this is also a overlap, some kind of overlapping, right? Pure, peak is this one, 29, 20, 19 is a very good peak. Even 30, you can see here, 30, you are looking at this particular image, you can get this is a smooth peak, but here this is a rough peak. Am I right? But if you increase the resolution, this may look different. That's why the right resolution is also very much important, right? In the tailing, common in real systems, it's normally, tailing is possible. If it is severe problem, if tail overlaps next peak or affect the quantification process. These are some types of chromatographic techniques. We'll discuss one by one. Since this is a certificate program, I cannot, in-depth explanation may not be there. I am covering all these chromatographic techniques. I will be giving you examples, some different case studies I have got from different articles. Okay, deep learning may be possible only if you learn different chromatographic techniques for longer duration, right? So the first one is paper chromatography. This is the first chromatography using a paper as a stationary phase. This particular chromatographic principles have been established. Then comes thin layer chromatography, gas chromatography, liquid chromatography. In the liquid chromatography, you have HPLC, right? Ion exchange chromatography, gel filtration, size exclusion chromatography, affinity chromatography. These are the commonly used chromatographic techniques in the food industries, right? Adsorption and partition techniques. In this, you have first one is paper chromatography. See, this is happening because of the adsorption, right? What do you mean by adsorption here? Absorption is different, adsorption is different, right? Adsorption is whatever the analyte particles adsorbing on the surface of the that particular carrier component. Okay, say in this particular case, paper chromatography, you have a stationary phase paper. These these analytes are absorbing on the surface of the paper so that you can clearly see the different colors, right? The solvent will move towards top, okay, over the stationary phase. You have kept your samples, okay. Say suppose for example, you can try at your home also. You take a marker, just point a drop on just the surface of the liquid, okay. It may be water also. Just put it on the water. The surface of the water should touch the dot. Slowly after few minutes, you can observe different color pattern, okay, like this, red color, purple, or different colors you can easily observe. Okay, then also we need to identify how far the solvent can travel, right? That is called as the solvent front. Say here only 10 centimeter the solvent will go travel towards top. After that it will not travel. Okay, so within that limit, how many different components can be identified, you need to analyze before going for this particular study, right? Stationary phase will be cellulose paper. Normal cellulose paper is also possible, water absorbed. Okay, water may be absorbed on the cellulose paper. Mobile phase, any kind of organic solvent you can use. Based on the partitioning between the phases, you can identify different pigments. This is called as the adsorption and partition techniques. What are the applications here? You can easily detect the dyes, right? So some color pigments are added into the food material. See here, it is very colorful, right? If you give these items to the kids, definitely they love eating all these, food items. Okay, so you can identify what type of food dyes are being used based on the absorption mechanism, right, using the normal paper chromatography. Qualitative identification and adulteration studies is possible. You can determine differences between permitted versus non-permitted colorants also you can identify. How this, if you are taking natural color, okay, the absorption phenomenon will be different, altogether different with the synthetic dyes, right? In that way you can easily identify. Thin layer chromatography, here it looks like this. Stationary phase is silica gel or alumina plate. Okay, so whereas in the case of paper chromatography, you used solvent, right? Mobile phase is organic solvent. Whereas in the case of this thin layer, solvent system is used, organic solvent can be used. Stationary phase will be silica gel or alumina plate. Visualization by UV light or chemical spray. Normally in the paper chromatography, you can observe in the normal light, okay, normal light. But whereas in the case of thin layer chromatography, using the UV light or chemical spray, you can observe what type of changes are happening on the paper, right? Adsorption phenomena can be observed only in the UV area, UV light. TLC applications in food analysis, you can see here, it is used for lipid profiling. Okay, triglyceride fatty acid profile, you can identify detection of aflatoxins, caffeine content, synthetic dyes. Okay, you can also identify preservatives and antioxidants. See in this particular picture, thin layer chromatographic profile using algal pigment. Okay, the chlorophyll A and chlorophyll B identification was done. If you place the algal material here, okay, and you just insert into the solvent, organic solvent, what will happen with respect to time? It travels, solvent travels towards the top. There is a different color absorption, will be there. Based on that, you can identify which one is carotenoids. Carotenoid color is different. Chlorophyll A color is different and chlorophyll B color is different. Based on that, you can easily identify. See here, this is the loading point. This is the point where it touches the solvent. Am I right? So sample loading point. So you can quantify how much you cannot quantify, but you can identify carotenoids are present in your sample. Chlorophyll A is present, chlorophyll B is present, right? This is how you can identify different kinds of pigments using the chromatographic, thin layer chromatography. The next and important principle is gas chromatography. See, the system looks like this. Carrier gas. Okay, carrier gas will be used. This is a mobile phase. Most of the time, majority 99.9%, it is inert gas. Inert gas is used. Why inert gas is used? Can anybody tell? In the gas chromatography, why inert gas is used? Anyone? Quickly.
So that it doesn't react with the sample which we are using.
Yes, very good, very good. See, normally if you purchase a packet of chips, nitrogen gas is filled, right? That what is the meaning of filling the nitrogen gas? To stop the oxidation process, right? Otherwise, what will happen? Oxygen molecules will start reacting with the potato chips and discoloration will be possible, right? So in that way, this inert gases, if you inject the inert gas, you use the inert gas as a mobile phase, it will not react with our sample. Okay. Similarly, in the chips also, nitrogen gas will not react with the sample. Okay. Then pressure adjuster will be there. Sample will be injected. Why pressure adjuster is required? Just like in the liquid chromatography, flow rate is important, right? Here in the gas, it's very difficult to find out the flow rate. So you will adjust the pressure of the gas along with the sample. It will be sent inside the column, and then that this is also based on the separation process. different molecules will get separated and it will start hitting the detector one by one. Based on the molecular weight, the peaks will be generated in the computer. That is the principle of gas chromatography. Separation, then those separated molecules will hit the detector, then that detector will convert that signal to the graphs. Graphs will be plotted. If you have a MS, mass spectroscopy in place attached with the gas chromatography, you can easily quantify how much quantity of the target substance is present in our sample. Am I right? So otherwise you can only tell this particular component is present in your sample, right? Mass spectrometry will help you to quantify how much quantity of this particular component is present in your sample. So the mobile phase is used is helium, nitrogen, hydrogen like this. Stationary phase will be liquid coated on column surface, right? This is the stationary phase in the column, liquid coated on column surface. You can see different columns are used for gas chromatography. This is the reason why liquid coating will be there on the column surface, inside the surface. Okay, different kinds of liquid coating is possible. Based on that, the separation will be taking place, right? This is the instrumentation of GC, carrier gas system injector. This is what happened. Column is there, hydrogen. Okay, this is the FID system. Okay, normally this is FID system is used. Detector will be FID, ions will be formed, and then this will be going to the detector, and it will identify what is the charge. Negative electrodes, if positive charge is there or negative charge is there. Okay, like that you can easily identify. Column packed or capillary column is used. Packed column or capillary column, you must have heard this one, right? In the research paper they will clearly mention packed column GC was used, capillary column GC, right? Detector, you have different types of detectors here. We will discuss one by one. Data processing is normal. See here you have these detectors. PDA detector is commonly used, photodiode array in case of this chromatographic principles. TCD detector, it is used for almost most of the compounds. Okay, most of the organic compounds. Detectable lower limits will be 10 ppm. Based on the detectable lower limits also, you need to use the detectors. FID detector is also commonly used. Most of the compounds including carbon at detectable lower limits of 0.1 ppm is used. Like that you have different types of GC detectors and mass spectrometry detectors. You can identify detectable limits, lower limits of the sample. Okay, this is how it looks. This chromatograph, it is the column is placed inside the oven. The oven temperature will be increased. Based on the increasing temperature inside the column, the separation will be taken place, and then it will be entering into the detectors, identifying and getting the peaks. Okay, so FID detector, flame ionization detector. Okay, this is the flame ionization detector. Flame ionization, because of the flame, ionization will happen, right? The ions will be traveling towards the detector. Then key detection principle, ionization of organic compounds in the flame. The main use will be hydrocarbons, organic compounds. Thermal conductivity detector, TCD detector. See here the short form is used. TCD detector, FID detector. Here I have given the abbreviation. Here you can see. Similarly, mass spectrometry. These are previously used detectors. Nowadays, everybody is using mass spectrometer detector. The key detection principle is like this: mass to charge ratio of the ions. Each and every chemical component has a different mass, molecular weight. It will detect the component based on the mass to charge ratio. This value will get as the result in the chromatographic principle. In the afternoon session, you can see mass to charge ratio, all these things. Main use will be structural identification and powerful identification via using the spectra. Spectra is nothing but the graph, right? Column types in the GC. These are different detectors you can use. Commonly used detector in the food system, I'm telling. Right? Different columns, packed column, capillary column, stationary phase column. Right? See, this stationary phase column can be used for polar, non-polar coatings. Capillary columns can be used for, this will be fused silica or high resolution. Okay. The packed columns will be stainless steel or glass. It has some kind of lower efficiency. If you want to just detect what kind of compound is present at lower cost, you can easily go for the packed detection. This is possible, right? Packed column. Capillary column is being used commonly. Okay. See here in the different types of columns, you can see the lengthwise the columns will be different. In the packed column, 1 to 6 meters. In the SCOT, different types of columns are there. These are the main difference is in the length of the column, inner diameters of the column, efficiency in terms of efficiency, right? And then sample sizing. These are some of the specifications for the column. Okay, you need to before going for the analysis, you need to select which column I need to use. Okay, next one is applications of GC in the food analysis, where you can use it for fatty acid methyl esters. FAM is profiling. For profiling you can use. Okay, then flavor and aroma compounds analysis, pesticide residues, alcohol estimation in the beverages. If the beverage is fermenting, then you can estimate the alcohol percentage in your samples. Characterization. This is example of GCMS I have given here. This peak is for the characterization of essential oil. You can see the HPLC retention time, I told you, you have a time here. These are the retention times. You can observe peak. Okay, based on the concentration, again the peak length will be there. See here, since this is the GCMS, all peaks will be red based on the molecular weight. What is the possible compound present in your sample? Even though if the peak is very small, you can identify. This is just for identification of the compound, not for quantification. You will identify that this particular sabinine essential oil is present. Then what you will do, you will purchase this particular standard, then you will analyze it separately, you will extract, and you will do the experiment separately to quantify how much quantity of sabinine is present in your sample. Similarly for other essential oils also. This is basic determination. For quantification, you need to again go for the different procedures, right? This is a case study given for GCMS to identify the pesticide residues in the fruits and vegetables, right? So here the extraction mechanism is Quetcher's method. Quetcher's method is used for determining the pesticide residues. Okay, Quetcher's method detection is GCMS. GCMS outcome quantification of organophosphate. This particular Quetcher's method you can usually remember. This is the extraction method used for GCMS for pesticide residues, right? In the second identification, you can see the flavor profile of coffee. You can see the aroma wheel of the coffee here. So many different aroma profiles are there in coffee. It's very complicated. In the fruity flavor, okay, fruity aroma, you have pear, and then different types of fruits, fruit related aroma. In the dried fruit, different types of aroma, sweets. Each and every aroma profile is mentioned in the aroma wheel, can be identified using the GCMS. GC olfactory plus GCMS. This is the different principle. Identification of the volatile aromatic components, aroma components, is possible using this particular method. Okay, this where you can use this one. This can be used for grading the coffee beans. Grade will decide the price of the coffee beans, right? So for this particular purpose, grading, you can easily go with the GCMS olfactory plus GCMS analysis for coffee. See here, extraction technique comparison. SP solid phase extraction and Quetcher's extraction. The basic difference, what is the purpose here? You can see the concentration and cleanup mechanism, whereas in the Quetcher's extraction, it is fast and multi-residue extraction. Comparatively, SPE, this is a little bit fast and user-friendly mechanism. Solvent used, how much, moderate quantity of solvent, very low solvent is being used. Steps here, you have different, comparison I have given here. Basically, Quetcher's extraction is, cost-effective process. You can see here in the cost, solid extraction is, phase extraction is costly compared to lower, Quetcher's extraction. That's why this particular extraction method is used for almost all the GCMS, analysis. Okay, this can be used for, complex food materials, plant components. Again, the, in the Quetcher's method, AOS International, they set the sample pH should be less than 5. In the European Union, EN, this particular method, they say that the pH of the sample should be 5 to 5.5. Okay, this is the main difference here. Liquid chromatography, the principle is same, where here you use the liquid, mobile phase. Separation is based on the distribution between liquid mobile phase and stationary phases. Okay, suitable for non-volatile and thermally unstable components. This is very important. If you are going for the volatile component analysis, then this liquid chromatography, this is not recommended. This is only suitable for non-volatile or thermally unstable components. Again, see, with respect to time, different inventions happen. You can go with the different detectors and different columns for estimating the volatile components, even for the liquid chromatographic as well. HPLC system components are like this, similar to GCMS. You have a injector, pump, solvent. Here the additionally solvent reservoir. You have a column, silica column or C18 column. You have a different detector, UV detector. What is the difference here? In this UV range, different frequencies, you need to analyze your sample. UV detector can be used, PDA detector can be used. Okay, different detectors we can use. We'll see one by one. Types of HPLC, we have 3 different types of HPLC. Normal phase HPLC. Okay, the column, it looks like this. The normal phase, reverse phase, HPLC, ion exchange. For each and every method, types of HPLC, column is different. Okay, in the reverse phase, you see non-polar stationary and polar mobile solvents can be used. Okay, these are HPLC detector, UV visible and PDA detectors for chromospheric components. Okay, if you want to identify chromospheric components, color related components, you can go with UV visible or PDA. Fluorescence, and this can be used for all the food, and PDA can be used for almost all 90% of the food. It is suitable. Fluorescence, okay, fluorescence detector can be used for identifying vitamins and polyphenols. Refractive index detector is commonly used for sugars, organic acid. Based on the refractive index, this particular detector can be used. Mass spectrometry, as you know that based on the mass, the identification will happen, right? For the structural elucidation. These are the applications of HPLC: quantification of vitamins and amino acids, determination of preservatives. If you have fruit juice, sodium benzoate, otherwise sorbate, you can identify these preservatives in this particular method, HPLC method. Caffeine estimation in the drinks is possible. And then phenol and flavonoid profiling. Like this, you will get the peak like this, holding time, different, this peaks will be formed, and based on the standard, you can identify at this particular retention time what is the compound. Okay, Here in this is the UV visible spectrometry detector is used. Absorption at 360 nanometer was used. Okay. This is how you will get the result in the HPLC. Later on, you have to process. This is the raw data. Collect the raw data. You can put it in the Excel to create a nice curves like this. Okay. Determination of caffeine in the beverage. Liquid extraction you can use for the determination of caffeine. C18 column is commonly used, and this analyze at 272 nanometer. Okay, you will get a peak like this, smooth peak you will get. This is the caffeine at 4.5, around 4.5 you will get the caffeine, right? This is the retention time. Vitamin C is also same thing. The retention time you can see here, L-ascorbic acid standard. Okay, you will identify standard first, then you will analyze the sample. This is for the reverse phase HPLC method. Okay, we saw different types of HPLC, right? Reverse phase HPLC method. The mobile phase used here is phosphate buffer methanol at 245 nanometer, right? 245 nanometer, you identify the L-ascorbic acid. This is the peak you will get, right? Later on, you can calculate the area of this peak and retention time. Okay, you got this at particular retention time. The area of the peak can be calculated. And aflatoxins also you can identify. Okay, what type of aflatoxin is present in the sample. Separation by HPLC FLD method at 365 to 455 nanometer. See here, aflatoxin G2 at this particular retention time. Aflatoxin G1 at around 10. Aflatoxin B2 at around 11. Like different retention time, different peaks you'll observe. That particular peak is correspond to what kind of aflatoxin we can identify. Quantify analysis at ppb levels. After calculation you can easily calculate, right? So how do you calculate here? You will know the area of the peak, right? Based on the area of the peak you can calculate how much. Why? Because the concentration of is directly proportional to the area of the peak. I told you in the earlier slides also, right? These specialized techniques, these are improvised techniques based on the ion exchange, right? This separation is possible. You can see here ion exchange chromatography. Sample will be injected and it will be burnt. Ions will be generated, right? This is the inside the column. You have this charges. The positive charges will be attracted to the negative charges. If the column length is 6 meters, 1 meter will be negative charge, 1 meter will be positive charge. Like that, different charges will be there. And based on the charge, the affinity will take place. Separation is based on the charge interactions used for amino acid profiles, protein estimation, peptides, right? This ion exchange chromatography you can use for protein. Nowadays, plant-based proteins, all the protein concepts is growing. So if you are interested into analyzing proteins, then you can go with the ion exchange chromatography. Stationary phase is cation anion exchange resins can be used. The graph you'll get will be looking like this, same. Okay. Size exclusion chromatography. Separation is based on the molecular weight. See, this is the stationary phase. Based on the molecular weight, the pigments will be trapped. Small, it's just like, pouring a, large gravels inside the beaker, then, putting the fine sand, then you put the water. The same mechanism is, observed in the size exclusion chromatography. The, based on the size, the retention will takes place. And this retention will also be, depending on the adsorption of the analyte with the stationary components. Here, larger molecules elute first. Okay, these are larger molecules. It will come out first. Okay, the application you can use it for carbohydrate estimation, polysaccharide, or protein fractionation. Okay, if you want to do protein fractionation, this is the best method. Size exclusion chromatography. Affinity chromatography, just like ion exchange chromatography, affinity chromatography. A protein has this structure in the column. It is like a lock and key mechanism. This particular gap, this will be used inside the column. When this protein molecule is entering, it will be binding. Okay, how much protein is being binding? So accordingly, we can calculate. Okay, not only for the protein, you can also use for different, purpose. This affinity chromatography based on the affinity. See here, this is called as the this is protein, and this is the ligand. Ligand, fixed on the matrix of the column. Inside the column, it will attach like this. Okay, just like an enzyme kind of, picture you can analyze. Used for enzyme, antibody, or toxin isolations. So many different types of applications are there. Supercritical fluid, SFC. Supercritical fluid, only the supercritical fluid is used as a mobile phase. See here, carbon dioxide is used. It is pumped. Okay. Instead of organic solvent, used in the HPLC, this particular supercritical fluid is used for chiral separation, lipid analysis. You can use this SFC for the lipid analysis. These are some of the advanced, combined techniques, right? GCMS, LCMS. This is, commonly being used. Okay, these are some of the advanced techniques. Instead of using only GC to identify, you can quantify using GCMS. Structural elucidation and quantification you can do. Method validation is very much important. As I already discussed, you need to validate the method. What kind of sample you are analyzing, how you are analyzing, using which instrument you are analyzing, what are the specifications you have to consider. Each and every parameter is very much important to validate the results, right? And one more important thing is, calibration curve, peak area versus concentration. Accuracy is important. Okay, repeatability is very much important. If you repeat the same experiment after validation, the result should be more or less same without any significant difference, right? That's why the result validation is very much important. What are some of the good practices in the chromatography is clean, sample cleanup, sample preparation, filtering. Basically, this is the sample preparation. This is very much important. Calibration and maintenance is also very much important. Calibration of the machines, what are instruments you are using. Maintenance of the HPLC or maintenance of GCMS, maintenance is very much important. Mobile phase separation and degassing. Degassing is very much important. If gas is trapped inside the column, then you will get a wrong results. That's why initial flushing is required. Data integrity and traceability. These are some of the good. So if you do all these things, what are the benefits? Enhanced data accuracy, streamlined result generation is possible. Okay, without any errors. Errors will be reduced. These are some of the challenges. What are the challenges? Matrix interference identification is important. You need to read more for selecting what kind of chromatography is required for analyzing your target component. Cost of the columns. Okay, same column cannot be used for all the application. Same solvent cannot be used. Chemicals are required, or maybe different columns are required. Sampling procedure will be different. Okay, time intensive method development. Okay, if you want to identify, you know uh, as a person, if I'm identifying, what is the retention time of glucose in the HPLC, that one is okay for me. I can easily do, and I can validate that one also. But in the lab, commercial labs, they have a validation protocols for so many analytes, right? That's why it is called as a time intensive method. They have to go for the calibration, they need to validate the results. It's a very Herculean task, and you need a skilled manpower for this particular chromatography. He should be able to read the results properly. So otherwise, what is the meaning of doing the chromatography, right? You will identify that this particular pesticide residue is there in the component. That is not sufficient, right? How much quantity is there, and what method you used, what are the specifications, each and everything, it will be read properly by the skilled manpower. You should be able to read the research articles, get the specifications. You need to analyze everything, right? These are some of the recent advances in terms of chromatographic techniques. Miniatured and microfluid liquid chromatographic systems are coming in place. UHPLC with sub 2 micrometer particles you can analyze. Previously, greater than 2 micrometer particles only can be analyzed. Now you can go with up to 2 micrometers or lesser than that. Portable GCMS systems for field use. Okay, unlike the traditional one, it occupies a lot of space in the lab. It requires air conditioning. It requires so many skilled manpower. Everybody cannot operate. So many restrictions. Nowadays, people are coming with the portable one. Then chemometric data analysis. Data analysis means what? Data analysis field is growing, right? Using Python or using machine learning tools, you can analyze the data. Instead of normally reading the graphs, you can import the graphs to the Python. It will read. You feed the mass. In case of GCMS, you just give these are the masses. The library. If you feed, then it will easily identify, give you the result. Crisp data for you required for publication or maybe identification. Artificial intelligence, machine learning are the main advancements in this particular area, right? Then these are some of the future trends. Integration of sensors, integration of cloud, okay, for saving your data, and the security, keeping your data safe is also very much important. Then real-time sensors with AI integration, all these things are very much important. Digital twin simulation for optimization. Earlier we used to do RSM optimization or normal optimization techniques. The optimization techniques are also evolving. So these are you need to learn, right? These are some of the key takeaways from this particular lecture. We have learned about what is chromatography, where we can use the chromatography, what are the different types of chromatographic principles are there, what are the different detectors we can use, what are the different columns we can use, where we can use, how we can use, how to read the graph. All these things we have learned, right? And it provides the sensitivity, selectivity, and reliability data. That also we have learned. Then support safety, authenticity. Authenticity again here for the profiling. If you identify any unique component, you can go for the publication, right? GCMS profiling of the particular fruit, new fruit, maybe fruits available in different districts which are not being explored. Advance continue towards the faster and greener techniques. These are considered as the greener techniques. Okay, these are the key things we have learned. In the afternoon session, you will learn how to do the experiment, how to read the result, data interpretation. You will learn, right? That's it for the session 1. If anybody has any doubts, you can quickly ask, because it's already 11:15. The next speaker is waiting for us.
Good morning, sir.
Good morning.
Thank you for the informative session, sir. Can you provide some insight on digital twin, sir? In the future direction, you mentioned about digital twin, sir. Can you tell me what the digital twin technique is?
Simulation. This is digital twin simulation for optimization. Means you will do the optimization, right? Normally, you will run different samples, 17 samples or 18 sample or 21 samples for the optimization. Then you will identify a single optimized result, right? If you go for the digital twin simulation, you need not to go for multiple experiments. You can simulate your experiment by doing very few experiments itself. That is called as the digital twin simulation. It is mainly used for optimization purpose. If you are going integrating with the HPLC, what you will get the result after 45 minutes, right? Initially 20 minutes runtime will be there. Then after that you will get 20 minutes for the analyzing sample. If you have 100 samples to be analyzed for machine learning process, at that particular time analyzing 100 samples using HPLC will be difficult. Am I right or not? So for that purpose, optimizing, extrapolating the results are very much important. You will get the simulated models, optimized values for getting your results accurate. So normally it reduces the errors. Okay, you will get the R square 99% or 99.9%. You can extrapolate the data and you can integrate this with any kind of analytical procedure to identify or maybe to know this particular compound is present. Okay, this can be integrated with the machine learning process. Is it fine?
Yeah, thank you, sir.
Okay, any other doubts?
Sir,
Yes.
sir, can you please give an example for pack column type chromatography?
What is that? PAP column.
Yes, sir.
PAP column, paper column.
So
What is a PAP column? I didn't mention any PAP column.
column types in gas chromatography, pack, capillary, and stationary phase you had mentioned, sir.
Okay, let me see that slide. Where is this?
Sir, I guess he is talking about the types of columns. The column
Okay.
chemistry, the packed column, capillary column.
Packed column chromatography.
Yes, yes.
Okay, normally see this different types of columns can be used, right? So in the packed column, normally solids will be used. Inside the column, you have a different, the column length will be 6 meters, right? So it's some solids will be used in the, from 0 to 1 meter. The the solid different in the packed column, normally solids is used. Some examples I told you, right? Silica, and then different solid materials we can use, right? Similarly, in the packed column also we can use it for fruit juice estimation, and any kind of juice estimation you can use packed column. So normally what the solid portions will be there inside the column. I could not find that in the side. Where can you tell? Yes, this one, right? Paper column, stationary phase. Are you clear? Packed column.
Yes, sir. Thank you, sir.
Okay. So nothing. It's made up of solid support materials. Okay. When it comes to packed column, solid supporting materials will be used for adsorbance. Okay, for adsorbance of your analyte. Nothing else. There are number of the adsorbance these solid materials are available. Okay. Okay, any other doubts? I think I will take the question and answers in the WhatsApp. In the meantime, we'll have the second lecture. Okay, it's already 20 minutes delay. Is it fine?
Okay, sir. Thank
Okay,
you.
Divya.
Okay.
Yes, sir. Thank you, sir, for such an informative and detailed session. So participant also inquire the question through the WhatsApp channel also. So now moving to the next session, spectroscopy. So spectroscopy is often described as the eye of analytical chemistry. So offering insight into the molecular structures and compositions, we are honored to welcome Dr. Arun Prasad Venugopal, sir, who will guide us through the principles, instrumentations, and applications of spectroscopic techniques. Sir is an accomplished academician and researcher with over 10 years of experience in teaching and research in the field of food process engineering. His expertise spans over the post-harvest handling and storage of fresh commodities, extraction of bioactive compounds, and design and development of food process engineering equipment. Sir obtained his PhD in Agricultural Processing and Food Engineering from TNAU Coimbatore. His scholarly and contribution over 30 research publications, 10 popular technical articles, and one authored book, reflecting his strong commitment to advancing research and education in food process engineering. Thank you, sir, for being with us. I request Arun, sir, to take over the session.
Thank you, Divya, for your introduction. Is my slides visible now?
Yes, sir, your slide is visible.
Yeah, I think we are good to go. So thank you once again, all the participants, for your patience participation in our workshop. During the second day, we are discussing about various high-end instrumentation used for analytical measurement and analysis. In that lineup, we have a spectroscopic technique. So this lecture will be an introduction to the spectroscopic technique and the spectrometry, about instrumentation and its application in various food analysis. So first, as we all know, this photo is light. Photometry is a measurement of the amount of light falling on a surface from a source. Similarly, spectrophotometry is the measurement of intensity of light at a selected wavelength. So the method depends on the light absorption property of either the substance or a derivatives of a substance that is being analyzed. So spectrophotometry use a prism, a grating for isolating a particular wavelength of a light. And this device allows us to pass the light over the substance, and there will be a receiver where it receives the light energy which scattered from the food materials or the samples which we are using. So now the spectroscopy deals with the study of interaction between the electromagnetic radiation and the matter. So the electromagnetic radiation, wherever we mention this electromagnetic radiation, we are talking about the light which is emitted from an electromagnetic radiation, entire stretch of electromagnetic radiation. I will explain you what is the visible range and which kind of lights falling within that electromagnetic radiation spectrum, where in which band of the spectrum the visible lights are there. So we'll explain that in later slides. So it involves analyzing the how different wavelength of electromagnetic radiations are absorbed, emitted, or scattered by a material. So it provides a valuable information about the composition, structure, and property of a matter. So as we all know, all materials are made up of atom and molecules. So when this electromagnetic radiation incident over a molecule, it causes some changes within the molecular energy level, and due to the absorption of electromagnetic radiation incident over the surface, and if you are able to trap the scattered energy out of the substances, we can able to make a molecular map or a fingerprint of a molecule, which shows us what kind of molecular interaction happening within the electromagnetic radiation and the materials. So to understand this spectroscopy, we need to understand nature of light. So electromagnetic wave are characterized by frequency and wavelength. Light is a spectrum of different wavelength, which our human eye recognize as white, but can be isolated into discrete portion by measured, discrete portion and measured. So the light which we are seeing in the white light which is visible to our eyes can be isolated into a discrete portion, and it can be measured using a device. So human eye responds to a radiant energy between 380 nanometer to 750 nanometer. But our instruments can measure shorter wavelength, and that is our UV, shorter wavelength, and longer wavelength, our infrared also. The wavelength describes a portion within the spectrum. It is the distance between two peaks as the light travels in a wave. So as you all know, the light travels in a wave motion. When you're measuring the distance between the two peaks, it gives the wavelength. The light is also composed of a distinct energy peaks called photons, whose energy is inversely proportional to the wavelength. So this photon, which is produced as energy, which is inversely proportional to the wavelength. So wavelength increases, the energy level decreases. That is what I'm trying to mention there. So basic concept of the spectroscopy, when light passes through a solution, a certain fraction of light being absorbed. This fraction is detected and measured using a light absorbance and transmitted to the concentration of the, and it is correlated with the concentration of the substances. So this enable us to qualitative and quantitative analysis of the substances. So what we are going to do when you're passing a light, a certain portion of the light is going to be absorbed by the solution, where another portion is going to be scattered. And if you have a device to catch the scattered amount of radius, scattered amount of light, and quantify the scattered amount of light when compared with the initial incident light, we can able to produce a map, the spectral map, which going to tell you about the interaction between the molecules, or it can be also be used to measure the quantity of such molecules present in the solutions and kind of molecules, what kind of molecule it is. So the spectroscopic technique is used to measure the light intensity as the function of wavelength. It is done by the following steps. Diffracting the light beam into a spectrum of wavelength. First, we need a light source, then it is diffracted into a spectrum of wavelength, and it is directed to incident over an object or our sample, receiving the light reflected or returned back from the sample, and detecting the intensity with the coupled devices, charged coupled devices, and displaying the results in a graph on a detector, and then it goes to the display device to show them the level. So the simple representation of our spectroscopic technique, spectroscopy can have a light source, and the light source is incident over a prism and gratings to monochromator, means which separates the light, which is a particular wavelength, which filters other wavelength, and it allows only a particular wavelength to pass through the sample. So the sample is placed in a cuvette where it doesn't interact with the light. It passes all the light which is incident over the surface, and then the sample which is holded in the cuvette will absorb some portion of the light, and some portion of the light will be scattered and pass through. So by using a simple photo cell, there are different types of detectors we will be discussing in the following slides, and the display meter. So the light absorbance is directly related to the concentration of the component in the sample. As the concentration of the sample increases, the light absorptions increases linearly, and the light transmissions decreases exponentially. So this is the key in understanding the spectroscopy. So when you're passing the light, when the concentration of the component, it can measure the concentration of the component in the solution. The concentration of the concentration of the particular solute increases, the light absorbance decreases, and the light absorption increases, and the light transmission decreases exponentially. So these are the wavelength with corresponding light, like 380 nanometers. Below 380 nanometers, we have ultraviolet radiation, which is in the visible spectrum, and then this 380 to 440 visible light, and up to like we have near infrared, mid infrared, far infrared. All these wavelengths can be used for spectroscopy and different instrumentations. So this is the entire electromagnetic spectrum, as you all aware. In an electromagnetic spectrum, we have a visible light region, and then like it starts from 100 nanometer, that is starts with UVC, UVB, and UVA, and then after the ultraviolet spectrum, we have visible spectrum. Okay, so it starts with gamma radiation, X-ray, and then we have yeah, just a minute. So this is the visible light region, and before visible light region, this is visible light region. Before light, we have ultraviolet range, and then we have red region. Beyond the red region of the visible spectrum, we have infrared. So that's why we call it as for the red region. So there, you can see the visible spectrum starts with the blue light. Sorry, the blue region. And then before that, we have a violet. So before violet region, we have ultraviolet region. Most of the visible spectrum, all the light from starting from the ultraviolet spectrum till this infrared red region can be used for spectroscopy. Even the infrared region can also be some portion like near infrared region. This particular region, near infrared region, can also be used for spectroscopy technique. Okay, so beyond this infrared, we have microwave and then radio wave. So this is the wavelength and the frequency. So when the frequencies increases, the wavelength decreases. So this is the relationship between the frequency and the wavelength of electromagnetic spectrum. So we can see the increasing wavelength here, like from gamma radiation and then the X-ray radiation. So we can, in the entire electromagnetic spectrum, the larger wavelength is radio wave. So when compared to the radio wave, microwaves are smaller. That's why it is called as microwave. Still, we have smaller infrareds are smaller wave, and X-ray, gamma are still smaller wave. Okay, so when compared to the larger radio wave, the next region, microwaves are smaller, and then infrared are much smaller. So this is about the electromagnetic spectrum. And then this some portion of this ultraviolet rays, ultraviolet UVA and UVB has some disinfectant effect, as it has a cause DNA damage. It can be used for disinfection also. That's why we use this UV light for disinfections in our laminar airflow chamber and everywhere. So now the electromagnetic spectrum. The spectrum is a range of all possible frequencies of electromagnetic radiation, as I shown in the previous slides. This energy associated with the electromagnetic radiation is defined as E is equal to hV. So E is the energy in joules, H is the Planck constant, which is 6.62 into 10 to the power minus 3.4 joules second, and V is the frequency in seconds. So spectroscopy allows the study of how matter interact with the emitted electromagnetic radiations. So as I mentioned in the previous slide, UV spectrometers are there. Near infrared spectroscopy techniques are there. So there are different spectroscopy techniques based on the light source which is used. So we can use the visible spectrum, visible light as well, and we can go for UV light as well. You can go for infrared red light as well. Sorry. Wavelength and frequencies, as I said, the relationship between the wavelength and frequencies. When the wavelength increases, the frequency decreases. The electromagnetic radiation can be considered as a combination of an alternating electric and magnetic field that travels through space in a wave motion. Because the radiation acts as a wave, it can be classified in terms of either wavelength or frequencies. We can represent in both the way, which are related by following equations, which is V is equal to C by lambda, where V is the frequency and C is the speed of light, which is 3 into 10 to the power 8 meter per second, and wavelength lambda is the wavelength in meters. So UV visible spectroscopy, the wavelength is usually expressed in nanometer, 1 nanometer, that is 10 to the power minus 9 meter. So visible UV spectroscopy, the lowest wavelength UV light has, and the UV light has a highest energy level. The energy may be sufficient to cause unwanted photochemical reactions, which measured during in the sample that are photosensitive. So when as we are using some visible light region and the UV light region, it may cause some damages to the sample which are photosensitive. In that, we have some more additional sample preparation techniques, and which requires a serial dilution or reducing the concentration of the component and reducing the light sensitivity of the sample. So how do we see the light? How human eyes see the light? Like when the light source is incident over the surface, the same happens. Based on the surface characters, certain portion of the visible light, as we all know, the visible light region comprised of so certain portion of the visible light absorbed, certain portion of the visible light, certain wavelength of the visible light region is get absorbed, only certain portion is reflected back. So we see only the complementary color which is reflected. The other portion of the visible regions are absorbed. So visible light region has all the colors, like based on the incident light. Most of the wavelength get absorbed, only a fewer wavelength get emitted out. So the complementary color with respect to the emitted lights is visible to our human eyes. So when electromagnetic radiation being incident over the surface or in a molecule, it causes some electronic vibrational and rotational transition within the molecules. So this transitional changes which occurs within the molecular level is observed, like it is measured in UV spectroscopy. So the water and then the carbon dioxide, all those transition of molecules and the energy level which occurs within the molecular level are measured. Now come to the transmittance and absorbance. As we are talking much from the beginning, this transmission and absorbance, let us see how it works. So this is the light from the light region. This I naught is incident light, initial incidental light over the surface of a sample. So usually the sample like it comes in a cuvette, in a glass or quartz container where the sample is placed, and in some of the portion of the light is absorbed and some portion is reflected back. And the reflectance, maybe we have a reference without any sample. So when comparing with the reference light and then the sample, which is the light which is scattered from the samples, we can able to measure the molecular interaction. When the sample is like when the sample is placed, it may absorb some motion or it may reflect back to some of the portions. So the transmitted light, that is IS, is a lower intensity than that of the incident light, I naught. And the transmitted light is defined as T, that is equal to IS, that is transmitted, absorbed, divided by the initial light. So to ensure the accuracy by eliminating the effect of reflectance by the surface and then the absorbance by the cell wall of on the solvent. So we need a reference. So that is what we are showing here when we are comparing it with the reference. So in the reference cell is inserted and the instrument is adjusted to give 100% transmittance, after which the percentage of transmittance reading is made on the sample. So the law which governs this absorption and transmittance changes is Beer's law. So this Beer's law states the concentration of the substance is directly proportional to the amount of light absorbed and inversely proportional to the logarithmic mean of light transmitted. So A, which is the absorbance, which is equal to absorbance of light, is equal to proportionality constant defined as absorptivity, and the light B, the light path in centimeters, and C, the concentration in gram per liter of absorbing component. So the absorbance A has no unit. So the unit of A is reciprocal of those B and C. That is our light path and then the concentration. So two components, two parameters will decide the absorbance. One is the thickness of the sample holder, or the thickness of the sample, the length, the length to which the light has to transmit it. And then the second thing is concentration of the sample, the concentration of the molecules which absorbs the photon component from the light. So when B is 1 centimeter, C is expressed in mole per liter, that is epsilon dash. is substituted for the constant A, and then the value of epsilon dash is constant for giving for a given component and given wavelength under prescribed condition of the solvent, temperature, pH, etc. So these particular parameters, solvent, temperature of the temperature during the measurement, and the pH of the samples, and the molar absorbivity epsilon dash varies with these parameters. So A, that absorbance is equal to log of I naught, that is initial incident light, divided by I, yes, that is I absorbed, is equal to A, that is proportionality constant, B, that is the light path, the distance of light path, and then the C, our concentration of the component. Okay. So, so this image you can understand when in the light source, incident light radiation, some of the sample absorbs some of the radiations, and then some few portion is transmitted and absorbed, transmitted and detected. Now, the application of Beer-Lambert law for quantitative analysis for sample preparations, and then the qualitative analysis for sample standard preparations, and then the quantitative analysis for measuring the concentration of unknown sample. So the absorbance, here you can see the absorbance increases with the constant increase in the concentration. So there is some linoleic acid concentration is given here in x-axis, in y-axis the absorbance. You can see the increase in the concentration with respect to the increase in the absorbance value. So spectroscopy nomenclature, as we discussed so far, like absorbance, absorbivity, molar absorbivity, path length, and then the transmittance, wave unit, absorption maximum. So these are some of the parameters we come across when you are discussing about the spectroscope techniques. So better to know about all those things. So in a direct proportionality between the absorbance and the concentration must be established for a given instrument under specific conditions. Okay, so the linear relationship exists upon certain concentration of the absorbance, beyond which the solution is said to be no longer obey Beer's law. So the basic phenomena for the basic thing is like the concentration of the solutions should increase with the absorbance. So Beer's law is followed only in the following conditions, like the incident radiation on the substance of interest is monochromatic. So we definitely need a monochromator or a filter which filters only the particular wavelength to incident over the surface, and then the solvent absorption is insignificant when compared to the solute. So when you prepare the samples, the solvent should be selected in such a way that the absorbance of light by the solvent is very minimum. The solute concentration is within the given limit. Beyond certain limit, there won't be any light reflected back or transmitted. So in such case, the transmittance will be cannot be measured. So solute concentration should be within the given limit. If it is beyond, we have to go for dilution techniques before during the sample preparation. And then the optical interference is not present, and then the chemical reactions does not occur between the molecules of interest and other solute or solvent molecules will be there. So the basic components of the spectrophotometers includes this light source, entrance slit or a monochromator, where the particular wavelength of light is filtered, and an exit slit and the cuvette, that is the sample holder, and the detector. So we may have a single beam spectrophotometer and a double beam spectrophotometer. The difference is the light source is entered into a particular slit after the monochromator, and then it is incident over the sample, and we have a detector to detect the the light transmitted through the samples. Whereas a double beam, we have a portion of the particular light is taken to a reference, and then the portion of light is taken to the sample. So we will be knowing the difference in the absorbance and the incident radiations. We have a reference to compare with the data. So we will have better results in such case. This is the schematic of a double beam and double beam in time spectrophotometer in space and time. So we have a light source and it enters into a mirror. Like initially this light source is entered and taken to a sample, and similarly the same light source is mirrored and entered into a monochromator with and it passes through as references. So this is double beam in space spectrophotometer and double beam in time. So the light after separating into a particular monochromatic light, then it is splitted into two of one is taken to the sample, another one is transmitted to the reference, and then we have a detector where it combines both the light which is coming in then, and then we go for a detection. So how does the modern UV spectrophotometer works? So ultraviolet, visible ultraviolet, the ultraviolet spectrophotometer uses the light source to eliminate a sample with a light across UV and visible wavelength, typically in the range of 190 to 900 nanometer. So the instrument then measures the light absorbed, transmitted, and reflected by the sample. So the some spectrophotometer have an extended wavelength range into near infrared region, that is 800 near infrared spectroscopy, where 800 to 320 nanometers. The wavelength range is higher in case of infrared spectroscopy meter. And then the instrumental design, as I said, it is very simple, which we discussed. We need a light source and then a monochromator, and then the sample, the provision to pass through the sample at the same time with the reference sample, and then the detector. So there are different types of light sources we have. There is no need to worry about much about this light source. Even whatever the light source, we have a monochromator to separate to filter out the other wavelength. So tungsten halogen bulb or xenon bulb, deuterium arc lamp, deuterium arc lamps are widely used as it is has good intensity within the UV region. And we have tungsten and halogen bulb, which is good intensity over the entire visible UV spectrum region. And most recently, this single xenon flash lamp is used widely, and then the xenon flash lamp is single source, has a significant advantage over the use of to conventional, like the other conventional lamps. So these are some of the specifications and advantages about using different types of lamp. Deuterium, that is D2 arc lamp. So this particular range 850, 185 to 400 nanometers, and low signal noise, and it can be the lamp has a higher half-life. The time required for the intensity to fall in half of its initial value, approximately 1000 hours. Similarly, we have tungsten filament, which is within the range of 350 nanometer to 300 nanometers, and it has a higher life of 10,000 hours. And the selection of the lamp depends upon the cost involved, and then the type of light, type of wavelength, or which portion of the wavelength is required. And in the xenon flash lamp, mostly the xenon flash lamp are not used in case of photosensitive materials. So mostly when you are measuring the pigments and light sensitive materials, we should not go for this xenon flash lamp, as it has photo bleaching effect. So so it is also having a wider range, like the one of the major advantages is it can be used for higher range, wider range, like 185 to 250 nanometers. So the next component in this list is monochromator. Monochromator is nothing, the different wavelength, it filters out the essential particular wavelength from the spectrum, entire spectrum. So holographic, holographic grittings, that is that small markings over the prism. So this is the gratings. You can see the small minute lines which allow us to separate the or split the incident light into different portion. So there are the multiple monochromators are used. The light source is passed through prism and there's the gritted surface. This gritted surface allow us to separate particular wavelength before incidenting over the sample. And then the dual beam spectrophotometer, as we discussed above, dual two light source can be used, or a single light source can be split into two different beams before incidenting. And then now come to the final part, that is detector. After incidenting the light over the sample and transmitted light has to be detected by some technique. So there are various techniques which allow us to detect the quantity of light transmitted. We have a photo multiplayer tube, which is commonly called PMT, which can be effectively used to measure 200 to 900 nanometer. So this is the schematic sketch of that PMT detector. So initially, whatever the light rays incident, we have series of mirror images, there are series of mirror, and which allow us to detect separate one particular photon, and it can be diverted to the detector. And then we have a silicon diode. Silicon diode can also be used to measure the it is also a kind of photodiode. The light fallen on the semiconductor materials allow the electron to flow through it and depleting the charges in the capacitor connected across the materials. So it is allowed to detect through a capacitors the incident, like the transmitted light is passes through it, and then due to capacitor it neutralizes the charges and it allows one photon to escape from this capacitance, and it can be measured easily. Lead sulfide detectors. This is another type of detector which detect the light within the region of 1000 to 3350 nanometers in higher range. And then the final element in the component is our cuvette. So cuvette is the materials sample holder, simply the sample holder. The requirement here is there are different materials used: quartz, optical glass, polystyrene cuvettes are there. They have their own wavelength suitable for a particular wavelength. The only requirement is it should have minimum absorbance. There should not be no absorbance. And then the cuvette cleanliness is very important in measurement. Presence of impurities, fingerprints over the cuvette has to be removed to ultrasonic cleaners, and then the chemical handling should be avoided. So even a small scratch over the cuvette can cause possible changes in the results. Post-blank measurement handling, like post-blank measurement is also very important to compare the changes. And then the thermostating the sample. Now comes to the sample preparations for spectroscopy. Thermostating is nothing, bringing the sample to a temperature where it can be effectively used for analysis. So in some samples, heating may require. In oil-rich samples, fat-rich samples, we may heat the sample, and in some case, like where we have to measure the volatile components, we will be going for cooling the samples. So there are different heating and cooling arrangements are there within the spectroscopy in advanced thing, and then stirring the sample. So stirring thermostated sample, like bringing to the particular temperature where it has maximum absorbance. and then stirring the samples to ensure the solution and the temperature is homogeneous. Into the entire cuvette, the solution has to be in the same concentration and the same temperature. So stirring is crucial for viscous sample and when studying chemical reactions in the cuvette. So maybe during some chemical reaction, what are the changes occurs during the reaction and what are the increase in the component, like formation of new component, increase in the concentration of the component during certain reactions can also be measured using spectroscopy. So the effectiveness of stirring depends upon the solvent, sample, and then the viscosity of the samples, and then measurement of low temperatures. There are some experiments, some analysis we may go for low temperature, especially when you are going for volatile component. So when you are going for this low temperature analysis, definitely we should take more care to prevent condensation by purging the sample compartment with clean and dry gases, and some UV visible system has purging port for the gas to enter without introducing light, before introducing the light to avoid condensation of the surface, and then the solvent transparency. So that solvent transparency is very important when you are measuring the liquid or dissolving solid samples for UV analysis. And the solvent are chosen based on the sample solubilities, stability of the solvent, pH requirement, and then the UV visible cutoff wavelength. So these are the essential things to be addressed before selecting a particular solvent. And moreover, the solvent should have least absorbance. So we go for aqueous, like water as a solvent. Like water is an excellent aqueous soluble component as it allows measurements through UV wavelength. Other organic solvents, they have certain limitations over UV range, and there are certain solvents which are performing well in infrared region with rather UV, and in UV most probably we go for water. So you can see this particular chart which shows the absorbance, this transparency range of solvents in UV region. So acetone has the least, and then the water has higher transparency, 100% transmittance, whereas acetone, and next to water we have isopropyl alcohol, methyl cyclohexane, ethanol, methanol. So all those things can be used, and even you go for hexane and acetone in most of the case. Acetone widely used in HPLCs and other chromatography technique where it has the because of its property, whereas water is widely used in case of spectroscopy technique. Stray light. This is another problem which arises whenever we go for spectroscopy. So apart from the light which is generated from the light source and the chromator, like monochromator, the influence of external light, that is the stray light, you can see the interaction between the stray light and then the absorbance. You can see the 1% stray light, how this curve deforms, and in the right hand side you can see a UV is system with poor stray light performance, which show the deviations of PS law, and then the make concentration calculations irreliable. So whenever there is a external light interactions, which always gives a wrong calculations. So linear range of instrument, like absorbance limit, higher absorbance issue, system limitations, and sample dilution, short path length of cuvette. So these are some of the problems which we have to avoid during this spectroscopy technique. Yeah. The wavelength are inverse centimeters, like most UV visible UV measurement are reported against wavelength in nanometers. In some older literature, you can often come across wave number. They use wave number 10 centimeter inverse. So wave number is often used in infrared spectroscopy, and the use of wave number scale is useful as it convey the change in energy level of the incident radiations. So lower wavelength gives a larger wave number and higher energy. So these are the functional group example, like and then there are absorbance range within the spectrum. So the application of UV spectrum in food analysis, determination of nutrient component. So we almost reached the final stage of the presentation. Like application, so far we have discussed about the equipment, the working of the equipment, principle behind the spectroscopy, and from this point we will be discussing about this what kind of analysis can be used. The determination of nutrient content, especially vitamins present in the food materials and protein present in the food materials, it has a very big potential applications. It helps in measuring the protein content using a specific reagents that form colored complex with the protein. So protein and nucleic acid melting temperature. So UV spectroscopy is commonly used in life science analysis for analysis of biomolecules such as protein and nucleic acid. So the UV spectroscopy exposes protein to heat or chemical denaturation. At certain temperature, the concentration changes in the concentration of the protein unfolds or melt and loses its structure during the spectroscopy. So protein at room temperature has a specific tertiary structures or conformation that in turn creates a specific electronic environment for aromatic and amino acids. For detection of additives and contaminants present in the food samples, like preservatives. So common food preservatives like benzonates and sulfates can be detected and quantified. Pesticide and residues present in the food materials can be detected and even quantified with this help of spectroscopy technique. So the concentration, again the same principle. The concentration increases with the absorbance. Yeah, quality analysis, especially in case of color measurement, detection of freshness and spoilage of food materials. So UV visible spectroscopy is used to assess the color of the food product, which is an important quality attributes. So this is the wavelength. Then the table behind me, we can see the wavelength and absorbent color with respect to the particular wavelength, and complementary color, which can be seen or reflected. Food authentication, like adulteration detection. We can use it as this particular UV spectroscopy having a higher application in case of detecting the adulterants present in the food materials. Origin verifications, like with respect to geographic origin, certain food product have certain particular components and unique spectral fingerprint. So this like especially this adulteration in honey, adulteration in case of coffee, adulteration in case of certain aromatic components can also be detected using this thing. Measurement of antioxidants. So polyphenols and flavonoids, and then antioxidant capacity, that is DPPH, which can be measured using this spectroscopy technique. In the absorbance and wavelength, you can see the particular DPPH in the 255 nanometers, the peak which shows the DPPH, and we have a DPPH control or standard solutions. When compared with the standard solution, the peak has the similar peak appear in all the samples, which confirms the presence of antioxidants in the samples. And the enzymatic activity, monitoring the enzymatic reaction, like enzyme inhibitors, enzyme assisted reactions can also be measured. Analysis of pigments, especially chlorophyll pigment, carotenoid pigment, with the specific absorbance range, like absorbance wavelength. Chlorophyll, you can see 430 to 470 nanometers, carotenoid 400 to 500 nanometers. So the peak shows the presence of certain pigment component anthocyanin 700 nanometers. When you have a peak at the 700 nanometers, it shows the presence of anthocyanin, chlorophyll, and carotenoid pigments. So these are the potential application where this spectroscopy technique can be used. So that's all from my side. So far we have discussed about application of spectroscopy. Before that, we have discussed about the instrumentation part and the principle and working of instrumentation. If you have any doubts related to this, please go ahead. Yeah. Participants, please, if there is any query related to this lecture, please go ahead. I think you can unmute and go for question. Divyadashni,
Yes, sir.
is there any question in the chat box? I cannot able to access this chat box from my side. I'm not able
No,
to.
sir, there is no questions.
Okay, fine.
Participants are like open for the questions and sir, there is one. From the participant
Participants,
side, if there is any questions? I guess no questions, sir.
okay.
Thank you, sir. Thank you for this comprehensive presentation over this spectrophotometric techniques. And we will now break for the lunch. And we will reconvene at 2 p.m. for the practical demonstration and result interpretation of chromatographic and spectroscopic techniques by Rashmi PK, research scholar. So we will meet there. Thank you.
Participants, please like try to be more attentive in practical sessions so that more than the theory portion, we highlight much more things in case of practical sessions. We'll also be there in the session. If you have any queries related to our lectures also, we can address during those presentations also. Thank you, participants. Thank you.
Thank you, sir. Thank you. Thank you all. We will meet again at 2 p.m. Thank you.
Ma'am, where we will receive the recordings?
Recordings will be shared at the end of the workshop only.
Okay, ma'am. Thank you, ma'am.
Thank you. Thank you.